Hello everyone, and welcome to our webinar today. My name is Christoph Meinert. I'm a co-founder and the CEO of Gelomics, and it's my absolute pleasure to have you all on board and listening to our webinar. So this is the first out of a webinar series of 3 this year that we are planning, so please make sure to stay tuned and subscribe, register to the future ones as well. And it's my absolute pleasure today to welcome Dr. and Associate Professor Jelena Kovacina today and have her present at this webinar today. So I'll just maybe start with a short introduction and a bio for Jelena. So Dr. Jelena Kovacina is an associate professor and an ARC Future Fellow leading a multidisciplinary group at the Graduate School of Biomedical Engineering at the University of New South Wales in Sydney, Australia. Her research interests are at the interface of biology and engineering, focusing on the development of biomimetic materials that direct cellular interactions for enhanced vascularization and for the treatment of cardiovascular disease. In particular, she develops novel silk-based biomaterials and investigates how biomaterial properties translate to biological outcomes. She completed her doctoral degree with Anthony Weiss at the University of Sydney and a postdoctoral training with David Kaplan's group at the Tufts University in Boston. She joined UNSW in 2014, where her research has been supported by funding from the Australian Research Council, the National Heart Foundation, the National Health and Medical Research Council, and New South Wales Health. She is a recipient of multiple awards, including most recently the New South Wales Ministerial Award for Rising Stars in Cardiovascular Research and the Early Career Researcher of the Year in Physical Sciences in the New South Wales Premier's Prizes for Science and Engineering. Jelena is actively involved in the Australasian medtech sector, including as a treasurer and secretary of the Australasian Society for Biomaterials and Tissue Engineering, the ASBT, who just recently also held a conference in New Zealand, and board member of the Australian Cardiovascular Alliance Bioengineering Flagship. She's absolutely passionate about research and training and communication, and I've known Jelena for about 10 years now, also met the first time at ASBT, so it's an absolute pleasure to have you here today. And maybe before we get started with your presentation, also just like to mention that obviously Jelena had a massive impact in the field of tissue engineering with over 60 papers published and over 4,500 citations. So really one of the future and emerging leaders in Australia and worldwide. So it's an absolute pleasure to have you here, Jelena, and talk about your research in biofabrication approaches, and particularly relating to silk materials. So before we get started with your presentation, I'd like to also point out to all the attendees that there is, of course, an opportunity for you to ask questions following the webinar. So to do so, please just use the questions or chat function in the GoToWebinar space, which I think you should find on the right-hand side of your screen there. So please feel free to ask anything you would like, and following Jelena's presentation, we'll go through those questions one by one. Unless I forgot anything important, I think we'll just get started with your presentation, Jelena. So again, thanks so much for joining us today, and we're really looking forward to your presentation today. Thank you.
Thank you so much, Christoph, for a very kind introduction, and thank you for having me here. This is a really cool opportunity, and I'm very happy to be here talking to Gelomics. It's a little bit of an unusual setup in that I'm talking into the abyss with the webinar system, but hopefully you're out there somewhere listening. So my goal today is to tell you, normally I might do a presentation that's around, here's the clinical need for, particular pathology, and this is how we're trying to solve it with our materials. But I thought I would do something slightly different today and actually tell you about how some of our recent work in silk crosslinking has actually enabled some really cool new fabrication approaches and new technologies that we're then applying towards those clinical problems and pathologies. So hopefully this will be of interest to the audience. Now, let me just make sure that things here work. Christoph has already done a very extensive bio and introduction, so I won't go into any of that. But I will say that my talk today will be almost exclusively about silk. I do work on other natural polymers as well, but I absolutely love silk. And I started working on silk when I was at Tufts with David Kaplan, and David sort of says, you either drink the Kool-Aid or you don't when it comes to silk, and I very much did. And so I've been at UNSW since 2014 at the Graduate School of Biomedical Engineering. Now, I'll start with a little bit of my sort of research motivation, what drives our lab, how we think about biomaterials research. So, you all know that biomaterials are materials designed to interface with the human body for either therapeutic or diagnostic application, and they're absolutely central in modern medicine, right? But if you actually look at most of these materials here in most of these devices that we're familiar with, they haven't actually changed extensively in a very, very long time, right? A lot of them were designed and are still being designed to serve a purely mechanical function. A piece of tissue is missing, we try to replace it, and the material serves a mechanical function. And then tissue engineering kind of came along as a way to address some of that, where the idea was not around that mechanical function, but really around regenerating tissue. And the standard concept of tissue engineering is obviously you take a material and you populate it with cells and you mature that in the body in order to replace an injured or missing piece of tissue or organ. However, while we've made as a field a lot of progress in this area, and especially towards engineering in vitro models using tissue engineering, for sort of clinical translation and replacement of organs, it hasn't come probably as far as we'd like, right? And this is in large part driven by logistical issues also. The logistics of the regulatory requirements, of the cost and the time involved in classical tissue engineering are really quite difficult. And so in the last little while, this idea of in situ tissue engineering has become relatively popular in the field. And so this is a slightly different approach where the idea is that you actually implant an acellular biomaterial in the body, just like those medical devices I was telling you before. But these materials are now designed to actually serve more than just a mechanical function, right? Where we accept that they actually encode quite complex information in their chemical and their biophysical properties. And that this information, these mechanical features, these morphological features, the pore size, the pore shape, the chemical structure, they can all be designed in order to promote appropriate absorption of proteins in the material surface, appropriate infiltration of immune cells that will actually serve a healing function, and then recruitment of appropriate cell types, vascularization, and so on. So the idea here that you implant an acellular material and your body acts as a bioreactor to get tissue regeneration happening. Now, don't get me wrong, this is not a trivial thing to do, but it is a nice goal to have when we design our biomaterials. And so a lot of our work kind of stems from this idea that we design materials believing that form can determine function. In other words, we ask, how do we elicit specific biological responses through biomaterial design? And so that's sort of the motivation that drives us. And all of this is in the context of cardiovascular disease for the most part. So obviously, I don't need to tell you that cardiovascular disease is the biggest healthcare burden in our society, and that there is a critical unmet need for new cardiovascular implant devices, including vascular grafts, cardiac patches following myocardial infarction, heart valve leaflets, and angiogenic therapy for ischemic disease. So we work across all of these devices to an extent, but with a big focus on biomaterials and how do we get materials that really integrate and function well through biomaterial design. And as I said, my favorite material is very much silk. Now, lots of animals make silk, including silkworms, spiders, bees, and so on. But the silk I will be talking about is silk from silkworms. So these, you see my hands actually here holding cocoons, and these cocoons are made as part of the metamorphosis process of the silkworm. So very briefly, the silk, the eggs hatch into the silkworm here, and as it goes through its life cycle, it actually spins a single fiber that it wraps itself around in to protect it from external factors while it's transitioning to this moth. And because it serves this protective function, it's inherently a very strong and very stable material. So silks are some of nature's strongest and most stable fibers. and a really interesting biomedical material. I'm going to dig into the silk structure just a little bit more because it becomes relevant to my topic today, which is how silk cross-linking is driving some of these approaches and materials. So bear with me while I sell you a little bit on silk. So as I said, if we actually zoom into this cocoon, it's made up of a single fiber, and that fiber consists of two main proteins, the silk fibron, and when I say silk throughout the talk today, I'm actually referring to silk fibron, which is structural component, and then there is this glue-like sericin coating the fiber, and as part of our processing, we actually remove the sericin. The fibron sort of arranges itself in a hierarchical structure, like a lot of structural proteins in nature. And if you zoom in some more, it's this very repetitive structure consisting of a lot of GA repetitive amino acids. And these arrange into these secondary protein structures called beta sheets. So these are very crystalline structures, very tightly packed together, held together by lots and lots of hydrogen bonds. And it is these secondary structures, these beta sheets, this crystallinity, that really gives silk its strength and its stability. Now, as part of our processing, we actually destroy those hydrogen bonds that hold these together. And so we break down the beta sheets, and that allows us to solubilize this fiber because it's not very useful to work with the fiber. You have very limited fabrication and material development options with the fiber. So we actually want to solubilize it. And we do that by breaking these hydrogen bonds, breaking down the beta sheets, and we get this soluble, solubilized silk fibron. So from this silk solution, now we can make all our various material formats that we like to work with in the lab that allow us to ask those questions about how form affects function. And I'm showing here only a subset of things that we work on, but really out there in the field, silk fibron has been made into a whole plethora of different material formats. Now, I kind of want to give you a little sense of why I like silk and what makes it such a uniquely processable material. So I'll give you 3 examples of how it, from the literature of how it's being used, all starting from the same building block. So our building block is this solubilized silk fibron, right? The solution of silk fibron, and we can electrospin that into these silk vascular grafts. These are from our own work in collaboration with Steve Wise at Sydney Uni. And so made entirely 100% out of silk, we can make it into these fibrous materials that are strong enough to be directly sutured onto a human vessel, human or animal, in this case vessel, and withstand physiological blood flow and pressure, and yet are compliant and elastic enough to match the properties of the surrounding vessel, which is very different to the current gold standard synthetic grafts made of synthetic polymers, which are orders of magnitude stiffer than the native vessel, which causes their failure. So we can make these strong but quite nice and elastic grafts. You can take that concept of elasticity and flexibility even further and make silk membranes that are interfaced with sensing devices to make silk electrodes for on-skin physiological monitoring. And these are really stretchy, really flexible, so that they actually conform to the shape of your skin and they move with your body, so you can go about your day while you're having physiological monitoring. Or you can take the same silk solution and go in a completely opposite direction, completely different, and process it into these tightly packed structures into bone screws, right? So these are now strong enough, tough enough to actually be directly screwed into animal bone. So from the same starting building block, through different processing and optimization, you can get materials that have vastly different properties and vastly different applications. And this is really why I like silk as a building block. It is very processable, very versatile, but it is also abundantly available, and it is a natural protein polymer, which means it is also biodegradable. Now, on to my main topic today, which is cross-linking. So when we solubilize our silk and then process it into these materials, such as these bone screws here, right, that would still be soluble in aqueous environments. So we need to reintroduce crosslinks in order to stabilize it. And traditionally, and what's really driven the field over the last 20, 30 years, has been the ability for us to reintroduce those beta sheets, to get silk to fold again, to reform those hydrogen bonds, and get that secondary structure that is found in the original native fiber. So these physical crosslinks, these beta sheets, are reformed in the lab, and that's how we get these nice and strong silk materials that have become so popular. And that all works really well for some applications. The trouble comes when we want to work with hydrogels, as many of us, especially in this audience, want to do, right? We want to work with hydrogels, whether for drug delivery or tissue engineering, biofabrication, tissue modeling applications. Unfortunately, silk hydrogels are opaque and brittle, and you can see this here. If you actually try to handle them, if you press on them, they really crumble. They're quite brittle. And so we never quite liked working with silk hydrogels. And so a number of years ago, we and others in the field started looking at alternative cross-linking methodologies, and dityrosine became quite a popular option. So dityrosine crosslinks are a covalent bond that forms between two phenolic rings that are found on the side chains of tyrosine amino acids. And silk naturally has a really good proportion, about over 5% of its amino acids are actually tyrosines. And this is really important because it means we don't actually have to chemically modify silk in any way in order to get these dityrosine crosslinks. Unlike your metacrylation, for example, that we have to do for a number of molecules, we don't actually have to modify our silk. And now these hydrogels that we make using dityrosine crosslinking are really easy to handle, very processable, quite elastomeric. Now, how do we introduce these dityrosine bonds? Well, you have a whole range of options, right? These include across in black here. If these are your sort of options, you can have a chemical pentane reaction that is sort of based on iron. You can have lots of options for photo-initiated cross-linking. So this is where you have a photo-initiator and then you add light, in most cases visible light, or you can have enzymatically catalyzed reactions that will cause that dityrosine bond. And the bond ultimately is, for the most part, the same across all of these. And the kinetics of the reaction can change, though, where the photo-initiated reactions are very quick, whereas the enzymatic reactions are quite slow. And what you see here on the y-axis in this table is kind of the types of fabrication approaches that have been enabled or interfaced with these different cross-linking technologies, right? So as you can see, for example, for ruthenium cross-linking, it has really enabled a lot of fabrication approaches, including not just casting of hydrogels, but sacrificial templating, extrusion printing, cell electrowriting, and so on. And so really, this dityrosine cross-linking, just like metacrylation has many years ago for silk, has now enabled us to fabricate materials using these modern fabrication technologies, including extrusion 3D printing, cell electrowriting, and lithographic approaches as well, but in the absence of any chemical modification of the base polymer, which is really exciting. And we can do that at fairly high resolutions, and you see lots of examples of this in the literature. The two sort of most common approaches for doing this, the most popular in the literature, are either via HRP, so hydrogen peroxidase enzyme, or via ruthenium SPS. So this is a photo-initiated reaction in the presence of visible light. And when I started working on dityrosine cross-linking, I was initially working with HRP cross-linking and didn't particularly love it. The reaction kinetics were very, very slow. When we tried encapsulating cells in these and cross-linking, the cells were settling down because it was taking so long to cross-link. And I wasn't really sold on this reaction. And then I started working with Kun Lim, who you guys would have probably heard from in one of these webinars in the past. And we started collaborating on ruthenium cross-linking, sort of photo-initiated cross-linking of silk. And here the reaction kinetics are much, much quicker. And it really then enabled us to do a lot of cool things through this dityrosine cross-linking methodology. And so we've recently went through a process of reviewing this in the field of how tyrosine cross-linking, methacrylation, as well as some plasticization technologies have really enabled novel silk material properties and fabrication approaches, which is now driving new applications. And that was just accepted and will be available very soon in the field. And we hope it's sort of a nice addition of how new sort of understanding of chemistries and cross-linking methodologies is really driving all these downstream processes. And what I want to do now is just give you a few examples of that from our own work, focusing on these dityrosine chemistries. So I have 4 examples that I will go through of our own research that was enabled by this new cross-linking methodology in our hands. And the first example is in collaboration with Kuhn, and it's around this idea of sacrificial templating to get nice, complex morphological structures in our materials. Now, sacrificial templating works on the basis of you interface 2 materials, and then you get rid of one of those materials, very simply. So in our case, we're 3D printing Pluronic. So we're 3D printing a material that you can 3D print very easily at very high resolutions, right? And then we're casting silk on top of that, and then we're removing Pluronic from that system. Now, you might be sitting there asking yourselves, well, why don't you print silk directly, right? That would make sense. And we can, and people do. However, we in our lab like to work with quite low concentrations of silk for sort of soft tissue applications, fast degradation, fast remodeling. And low concentrations of silk typically do not have the appropriate viscosities necessary for that good printing window. And often in 3D printing, you have to actually be optimizing that printing window at the expense of your ideal material properties. And so for that reason, we're actually big fans of sacrificial templating, where you print a material that prints easily, and then you cast your material of interest on top of it. And so we wanted to ask whether we can actually get high resolution structures by sacrificial templating silk on top of Pluronic. And we tried this using both the enzymatic approach, using HRP cross-linking of silk, and the photo-initiated approach, where we're using that ruthenium cross-linking of silk. And as you can see, in the presence of ruthenium photo cross-linking, because it happens very quickly within 30 seconds, we can get very nice high resolution structures in silk on that Pluronic, whereas with the enzymatic cross-linking, we're not getting that because the reaction kinetics are so slow. And we can use this to get some really intricate structures, and we have lots more examples in this publication of these, but you can see that using this very simple approach, we can get fibrils of different sizes, we can get different pore structures, shapes and sizes, different spacing between these fibers, so you can get high resolution intricate structures using this relatively simple approach. And the photo-initiated cross-linking also allowed us to encapsulate living cells at very high cell density, so about 15 cells per million, unlike the enzymatic approach, where the maximum was about 2.5 cells per million. And again, this is related to those reaction kinetics. And we did this for sort of chondrocyte encapsulation and have published on that cellular work. I won't go into it today. But where my interest with this technology was now asking, can we use these same structures then, this same technology, to make these different structures, and ask how that affects tissue biomaterial integration in vivo. And so we use the same approach, print out Pluronic, cast out silk, cross-link, get rid of Pluronic, to make 3 types of materials out of silk. We made materials with no channels. We made some materials with just parallel vertical channels that are about 500 micrometers in diameter, running in one direction, and then some channels that were interconnected, running in both directions. And then we lyophilized these to give them porosity in the bulk as well, not just through these channels. And then we implanted these in vivo, just subcutaneously in mice, and looked at the percent tissue infiltration into these. And no surprise here, right? This was not a surprising result. We and others have shown in the field that having microchannels actually allows more tissue infiltration and better integration of materials with the body. So this wasn't surprising. But what was a little bit surprising to us was when we looked at the sort of the foreign body response and the fibrous encapsulation of these materials, which was quite different. So with our node channel scaffolds, even though they have porosity, we were getting a little bit of tissue ingrowth here into the periphery of the scaffold, but the scaffold was also getting encapsulated by this fibrotic capsule. And this is not unusual. A lot of materials, when you implant them in the body, this is a typical response, right? Macrophages cannot phagocytose these materials because they're very, very large. And so the body encapsulates them in layers of fibroblasts and collagen to sort of isolate that material as a foreign body. With our parallel channel scaffolds, we're getting more tissue ingrowth and a little bit of a thinner fibrous encapsulation response, but it was still there. But with our interconnected channel scaffolds, remember, we're just changing the morphology here. The material is the same. We were not getting that response, or very little of that response. And so what this was telling us, that by simple material design, by simple changes in material design, we're now affecting this very complex foreign body reaction to our materials, and is encouraging for us to think about material design as a way of actually controlling complex biological processes. And it was really enabled by this ability to cross-link silk now into complex structures in the absence of having to optimize all the 3D printing technologies and so on. All right, so my second example is around capturing another type of complex tissue morphology, which is alignment and anisotropy in mechanical and morphological properties. So a lot of tissues in our body, including, for example, skeletal and cardiac muscle, neural tissues, ligaments, tendons, and so on, have inherent alignment in their cells and extracellular matrix. And this alignment is absolutely essential for the mechanical functions of these tissues, but also for solute transport and for propagation of electrical signal, for example. But it is something that's not trivial to capture in three-dimensional scaffolds necessarily. And we've been working on this for a little while and are quite interested in this technique. And we do this using a process called ice templating. Now, this is an old material science technique, and nothing particularly new here, but I'll tell you how we're interfacing it with tyrosine crosslinking, which I think is a little bit interesting. So let me just give you the basic principles. So we start with a polymer solution, in our case, silk in water. We freeze it, and that causes ice crystal formation. And then when you remove those ice crystals, everywhere you had an ice crystal, you now have a pore. And by changing the direction of the freezing, you can change the shape of those pores. So if you freeze in a regular freezer, freezing will happen from all directions, and you will get mostly rounded ice crystals and pores. And so you might get a scaffold that looks like this. This is our silk. And this might look like the extracellular matrix of skin, for example. Whereas if you change the freezing, so it can only happen from a single plane, single direction, the ice crystals will start growing in a very elongated, aligned manner, and you're going to get these aligned pores. And this is now starting to look like the extracellular matrix of something like skeletal or cardiac muscle. And you can change the size of those pores by changing the freezing rate. So as a general principle, if you freeze faster, you get smaller pores. And you can obviously engineer pore gradients. These are pore gradients in our silk scaffolds through the same process. And we have this ice templating setup developed. When we were developing it, it was based on some design criteria that around we want it available on a lab bench. We want it really versatile and that can be transported and available to non-engineering labs as well, but with a decent amount of control. And we're very happy to share these designs as well as to ship some of these out to people. But we can now use these to make really nice, large, unidirectionally frozen aligned scaffolds. But what happened during this process when we were studying this freezing, an interesting thing we noticed. And so bear with me for a second because it's a bit of an unusual story, and I'll get back to hydrogels. It's related. So normally, as I said, if you slow down freezing, you get larger pores, and we're indeed seeing this. This is the pore area here. This is the silk concentration of different silks, right? And with the mesh insulator, we can slow down freezing. So each time if we slow down freezing, we see larger pores. But the 10% silk concentration, we're seeing these absolutely huge pores. Very, very surprising relative to 4 and 6% silk. and yet the cooling rate, the freezing rate of these is the same. So it's not related to this well-known process of slowing down freezing. So we started exploring what might be happening here, and we had this hypothesis that it's to do with the fact that 10% silk is actually quite viscous, and that silk is a shear thinning material. So if you increase shear on it, its viscosity reduces. And so we think when we were freezing at these uninsulated fast freezing conditions, the ice crystals growing through the silk were actually causing shear, and that was causing lowering of the viscosity of silk, and that was allowing it to actually freeze in a sort of typical manner. Whereas once we slow down this freezing, there is less shear, silk remains viscous, and these ice crystals can keep displacing the polymer and growing, right? But this was, we could show this, we modeled what those freezing conditions are and what the shear rates applied by the ice crystals might be, but it's still a sort of a difficult thing to prove. And it was this cross-linking of silk that allowed us to actually take this a step further and prove it, because through cross-linking, we could introduce sort of artificial viscosity in low concentrations of silk. So we did not have to work with these 10% silks. We could work with low concentrations of silk. So if you take your silk solution at low concentrations, like 2%, you don't get very high resolution pores. You get these sort of very fibrillar structures at very low concentrations of silk. However, if we now take our cross-linked silk, whether through beta sheet or through dityrosine bonds, and now freeze it, we get very large pores, very high resolution pores, because we have now again artificially introduced this viscosity, in inverted commas, into silk, right? And so it is now, this dityrosine cross-linking is also allowing us to ask these fundamental questions about the freezing processes and thermodynamics and the interface between that and material properties, which I think is really exciting. But that's not the only reason we freeze hydrogels. We also freeze hydrogels because hydrogels have nanoporosity, and freezing allows us to introduce micron scale porosity into these while maintaining some of those really cool hydrogel properties, right, that they're known for. Also, hydrogels allow us to incorporate lots of substances, including in complex manner, right? We can add molecular gradients and patterns of molecules in hydrogels that we cannot do in silk solutions when we're freezing them, right? And I'll show you an example of that in a second. But when we started freezing hydrogels, we actually had to optimize our cross-linking again so that we can be freezing directly in these molds that we ice template our silk in. And that took a little bit of optimization because our normal optimized cross-linking conditions of ruthenium and SPS for initiator were not actually working. We were getting lots of uncross-linked silk. So it took a few months to actually optimize this, but we can now make these very large constructs that we can take out and manipulate if we need to, not that we do, but really nice cross-linking in this system. And we can ice template these, right? So we can now unidirectionally freeze these hydrogels, and we get alignment in our pores and interconnected pores. And we know we have anisotropy in mechanical properties. So depending on whether we pull in one direction with the pores or against the pores, we get different mechanical properties, telling us that we do have this inherent anisotropy in our materials. And these retain the inherent properties of their original hydrogels, right? Where these are now ice templated, and the ones that were beta sheet cross-linked actually are still brittle and difficult to handle, but the ones that are dityrosine cross-linked and now we have porosity introduced into them are actually really easy to handle, quite elastomeric. They recoil really nicely. And because they're so easy to handle, it means that we can start processing them using sort of standard processing techniques, including cutting them with the vibrotome without the need to bed them or process them in any way. So we can cut them down to 200 and I think now even 100 micron thick, so that we can implant them or use them to grow cells on and so on. But as I said, to me, the coolest thing about these is that we can now start introducing concentration gradients into these very large structures. So this is now a nice templated silk hydrogel where we've introduced two different molecules, and you can see you can get molecular gradients and these interfaces in the materials. And these are now trapped in there, which we could not do with our solutions. And because we're doing this via dityrosine crosslinking, it means that if any of these molecules are proteins, most proteins have tyrosine amino acids, which means we can covalently trap them in the hydrogel. So Zach has shown this with BSA, for example, that if we encapsulate BSA in our beta sheet cross-linked hydrogels, it is released very, very quickly. Whereas if we trap it in our dityrosine cross-linked hydrogels, it actually stays in the hydrogel for a long time. Even this is after photo cross-linking and ice templating. And then there is the obvious advantage of the porosity that you've introduced, where your hydrogels have nanoporosity. So when we implant them in vivo in mice, so this is under the mouse skin here, you can see a little bit of tissue growth, probably fibrous encapsulation around the periphery of these, but there is very little to no tissue growth into the actual hydrogel because it takes a long time for the enzymes to degrade silk and actually grow in there. Whereas with the lyogel, we're getting really good tissue ingrowth into these materials. My last two examples will be very quick. The silk microgel work is again related to this issue that we have, that we like lots of things about hydrogels, but they have nanoscale porosity, which makes it really hard for cells to actually modify them and change them and grow into them, and for vasculature to grow in. And so one way to solve that in the field is through microgels, creating microgels. So this is where you create tiny little hydrogel spheres. And in our case, we make them using a microfluidic device through phase separation, and we cross-link them using dityrosine cross-linking. And because this reaction with ruthenium is so quick, we are actually able to capture these little microgel spheres as they're coming off this microfluidic device here. But the cool thing about these is that you can now start packing them together, and because they're rounded, they're not going to pack perfectly. And so between them, you're actually going to start getting porosity. And so now you have all the properties of the hydrogels with the porosity, with the micron scale porosity between them. And you can make them into these scaffolds like here. But the other really exciting thing about these is that your regular bulk hydrogel is homogeneous, right? So you have the same properties throughout the whole material. Whereas you can make these heterogeneous. You can make a population of microgels that's loaded with, say, cell 1 or molecule 1, and another population that's loaded with a different cell or a different molecule. And you can start packing those together at different proportions in order to get more complexity in your tissue engineered constructs. And when we take these and we put cells in them, if you encapsulate cells in vitro in these, you can see live cells staying green. And in our bulk hydrogels, they're happy enough. They stay alive for 7 days, no issues, but they're not exactly spreading and they're not proliferating much. Whereas if we encapsulate them between our microgels, those cells start off rounded and then they start spreading and proliferating and really modifying their environment. And we see a similar effect in vivo. If we implant these, the bulk hydrogels get fibrous encapsulation very, very quickly, where you get this collagen deposition in blue here. But the microgels are getting very little to no fibrous encapsulation, and you're getting all this tissue growth between the microgels, and you get blood vessels growing between them, and you're getting remodeling of this matrix. Okay. I will take another minute and give you my final example, which is slightly unusual. It's not really about fabrication, but it is around biofunctionalization of silk. So we work with lots of materials. I've been selling you on this idea of dityrosine cross-linking, but we actually work on lots of materials where we like beta sheets in our silk. We've optimized the materials for beta sheets, and we like working with them, and we don't want to change them. And an example of that for us are these vascular grafts that we've been developing with Steve. And this is for sort of small diameter bypass grafting, or so coronary or peripheral applications where you want to bypass occluded vessels. And it took us a long time to actually optimize these low porosity and high porosity grafts that we've now taken through a series of animal studies in rats, and are currently implanting in sheep. And so we're happy with these. They're performing really well. They're outperforming their synthetic counterparts. And so we don't want to be changing their mechanical properties, but we might still want to be immobilizing certain proteins on the surface of these, or certain molecules. And so we think we can still use those tyrosines that are available in silk, even though it's beta sheet cross-linked, in order to immobilize another protein on the surface. And so we have shown this in another set of studies where, we can now take a pre-established silk material that has these tyrosines available naturally in it, and any protein peptide that has tyrosines, and using those same chemistries I've been telling you about, you can now immobilize these molecules just on the surface of silk. And we've shown that with an example by immobilizing perlecan, which is a vascular basement membrane proteoglycan, where this is perlecan absorbed, passively absorbed on the silk materials, cross-linked, immobilized via EDC and HS chemistry, which is a very common chemistry used for silk, or enzymatic or photoinitiated dityrosine cross-linking. And so you can see that we get similar amounts. But if we actually look at the amount that is lost following strong detergent washes, right? So how much is lost? How much is not covalently bound effectively? We see that with adsorbed, pretty much everything is lost because there's no covalent link. With EDC and HS, we lose less, but with dityrosine cross-linking, we lose very little of the protein. And that makes sense because we have more amino acids available for this dityrosine cross-linking than in the reactions involved in EDC and HS. And so with that, I will finish up. And thank you very much for your attention again, and I'd be happy to answer any questions.
Thank you, Jelena, for a fantastic presentation and really very impressive work there as well. So to all the attendees, please feel free to ask any questions that you might have. I might just kick off with one of mine as well, Jelena, if you don't mind. Got quite a few actually, so might just get started and get the ball rolling there if you don't mind. So silk seems to be a really fantastic material, and it's been used also particularly by your group in Australia in the tissue engineering field quite extensively now. And I can see there's a lot of new application coming through that look very, very promising as well. I was curious whether silk can also be now produced in a recombinant manner.
Yes, so there's been for a long time there have been a lot of efforts to make silk in a recombinant manner. Most of the work has focused on spider silks, just because silkworm silk is so abundantly available, whereas spiders are predatory. So those silks people have focused on a lot for recombinant production. And there has been an effort a long time ago now to actually make a transgenic goat that will make silk in its milk and to purify it that way. I don't know what's happened to that effort, but definitely it's an option. Silkworm silks they have been made, but there are some difficulties because it's such a repetitive molecule. They do present a little bit of a challenge for recombinant expression.
Fantastic. Thanks for that. So from my understanding, from how you answer this question right now, there is differences between different species of animal as well in terms of the composition of the silk. Is that general amino acid sequence changes, or the relative contribution of a certain amino acids in the overall chain?
Both, I guess. There are lots of things about the overall sort of structure and the patterns in silks that are conserved between species, but there are definite species differences, especially in amino acids that are used. So, for example, spider silks have more alanine, as an example. But there are also differences between species. So even within silkworms, to give you an example with tyrosine, the domestic, so the Bombyx mori silkworms we work with have about 5% of tyrosines, but they naturally don't make a dityrosine bond. Whereas there are wild type silkworms that actually do make these dityrosine bonds and have very different fibers as well. And so, it becomes this question of is it the evolutionary pressure or the environment? What's caused this?
Oh, fantastic. So there's quite a bit of interest around the dityrosine cross-linking, and in particular the photocross-linkable one. I also have a few more questions, but I can see there's a few from the audience as well that I might just go through one by one if you don't mind. One of the questions is, how many minimum tyrosines are needed in the protein for dityrosine cross-linking? A bit of a tricky one.
that becomes a chemistry question, and this is a Kuhn question probably more than me. But I don't actually know the minimum number, but I know that people do make tyrosine-rich peptides, so short peptides that are based on tyrosines. And people have made silk elastin copolymers with tyrosines. And so at least with those short peptides, I think they have one or two, and then the next molecule. So for intermolecular bonding, you can do it with very few. But what we don't know with our silk is how many are available. This is very much currently a shotgun approach. But we'd like to get a little bit more specific about how we do the cross-linking.
Absolutely. But it is generally possible to functionalize proteins with tyrosines. Is that correct, from your understanding?
Yeah, you can add tyramine residues, and people have done that. Yeah.
Yeah, so essentially, if you come across a material that you would like to cross-link via dityrosine cross-linking, and it doesn't have any natural tyrosine in it yet, it is possible to add it via functionalization.
Oh, absolutely.
Yeah, next
and
question.
People have actually done. People have added more to silk as well to increase the cross-linking efficiency as well. Yeah.
Fantastic. So I might just move on to the next question. The next question is surrounding if silk will dissolve. Which solvent is used for the dissolution of silk, and what type of concentrations can be dissolved or achieved?
So I assume this is for the original fiber.
Yeah, that's my understanding as well.
Yeah. So pretty much, there are a number of strategies, but the most common ones are around high concentration salts, and we use lithium bromide, specifically at 9.3 molar. That one I can answer. But there are other calcium-based salts and so on that are used for dissolution of silk. There's actually a really nice review on that I'd be happy to share with people. But our favorite is lithium bromide. You want a high concentration salt that's going to break those hydrogen bonds that hold it together.
Okay. So you're disrupting the hydrogen bonds, and that will open up the beta sheet. So
exactly.
Does that mean that
you dialyze the salt out?
Does that mean that during the processing of the solubilized silk, you have to have the salt present at all stages, or can you remove it later?
No. You can. We definitely remove it. So we solubilize it. We dialyze the lithium bromide out, and as long as it's in solution, it is stable for a little while, depending on the concentration. So we tend to keep it for about a month, and then it will naturally form beta sheets, and it will naturally gel actually.
Oh, fantastic. Cool. So next question would be whether pH and the solvent play any important role in dityrosine cross-linking, and whether the tyrosine cross-linking should be performed in an acidic, physiological, or alkaline environment.
That is a great question, and the answer is probably more complicated than the person asking has anticipated. So the simple answer is that we mostly do this in physiological pH, right? So we tend to either work with sodium chloride at physiological pH or in water, and that seems to work, and we don't seem to have a big difference in this. But an interesting thing happens with silk, right? And we have a whole project on this, in that once you dityrosine crosslink it, most sort of polymer chemists would kind of tell you it's stuck there. You have a covalent bond, it's stuck, right? But we find that locally silk can still change and form beta sheets in certain regions. And so you can get this stiffening of these materials over time. And that very much depends on the original cross-linking conditions as well as the environment it sits in. And so pH seems to play a role in what pH you crosslink under, and we can seem to be able to crosslink under all these pHs. We've done a range. But how stable that material is going forward is affected by the original cross-linking pH.
Okay.
Why? I don't know. We're figuring it out. But the environmental conditions do seem to matter.
It's an interesting observation. So we have one more follow-up question from the same attendee. If dityrosine cross-linking can also be performed on, I guess, native beta sheet silk, and whether it's improving or changing the mechanical properties.
Oh, that's a great question. I don't know if you could actually dityrosine cross-link an already beta sheet containing material. Maybe. I don't know. That's interesting. I'm going to make my students do that experiment now, actually.
Fantastic. Look, I've got a few more questions as well. So, especially with dityrosine cross-linking, my understanding is that some proteins, of course, have tyrosine groups naturally occurring in the amino acids sequence as well. So I'm curious whether you've investigated adhesiveness to tissue, particularly when you cross-link in situ or in an in vivo environment, let's say. Does it adhere to the surrounding tissues as well?
Yep, it definitely does. We have. I know Pruden has already published on this, I think, for gelatin, some form of modified gelatin, and through dityrosine cross-linking adhesion to the tissue. And other groups have done a little bit of that as well. I think there's a group here in Australia that have, I think it came originally from some of the work from CSIRO that actually did
so gelatin sealant,
the material that they did, but dityrosine cross-linking to the tissue. Specifically for silk, we have also, we definitely find that it will adhere to, we've done muscle, we've done lung, a bunch of tissues, yeah.
Fantastic, that's really good. And in terms of your hydrogels, if you cross-link them with also dityrosine cross-linking again, we've talked about adhesiveness now. I'm curious in terms of mechanical properties, you showed some effect of increasing the toughness of the material, which I think is one of the beautiful things about the dityrosine cross-linking. Do you also see with silk these super stretchable gels once they're cross-linked with dityrosine?
We do. We see more stretchability relative to beta sheet, which are not very stretchable at all. So they are definitely stretchable. I wouldn't call them super stretchable necessarily, because silk is quite a tough material. Super stretchability with silk seems to come from the introduction of calcium ions and plasticization of silk. And there's been some beautiful work done on that in sort of on skin monitoring devices. And we are quite interested in actually interfacing now this dityrosine cross-linking with calcium ions and plasticization, and looking at how the two work together. But that's very much, at the very early stages of work.
Fantastic. There were two more questions. So regarding the BSA release assay, I think you're showing the release of BSA from dityrosine cross-linked silk hydrogels was significantly slower than from a beta sheet. And curious about the mechanism, if you can comment on that. So is the BSA being cross-linked as well through dityrosine? So it's a covalent cross-linking there?
Yep, that is our assumption. We know that BSA has tyrosines in its amino acid structure, and we know that the release profile is so much slower than with beta sheet. So our assumption is that it is actually physically cross-linked in there. Now, it could theoretically be more trapped in there based on the sort of the mesh structure of the hydrogels. But when we look at the mesh structure between beta sheet and tyrosine crossing, I don't think they're different enough to actually be the reason for this, to be the explanation.
I would also agree. I think that seems to be the most plausible mechanism there as well. I'm also curious with the dityrosine bonds again. Would you expect hydrolysis to occur over time that could further release BSA and other protein molecules from this, hydrogel as well?
So that brings us to a bigger question, which is that we don't know how these will degrade, right? And that's something where, and when I say degrade, I mean in vivo, because silk by hydrolysis does not degrade all that quickly, right? And it's mostly you want enzymatic MMP-based degradation. And so we're, as we speak, implanting them in vivo to look at their degradation profiles, because we know them quite well for beta sheet-based materials, but not for these dityrosine cross-linked ones. So, and that will then ultimately affect also BSA release.
All right, so last question we had was regarding the microgels. So I thought that was also really, really cool, looked really nice pictures there too. And I'm really curious, we've got a question there, how the spreading cells can be explained. Is there a functional, I guess, biological activity Or do you think there's absorption of BSA or maybe some molecules from the surrounding FBS where the cells then attach to?
Oh, in terms of what's actually allowing the cells to grip and attach, I think it's definitely the silk has no, a Bombyx mori silk has no inherent cell binding domains, and so all the cell binding is either through, you know, a non-specific sort of electrostatic type interactions, or more likely that proteins absorb from FBS. And we definitely see that when we, you know, grow cells on silk, we do a lot of sort of adhesion studies. Without functionalizing silk, we're getting no cell interactions. If you functionalize the surface, we get really good interaction. So I'm 100% sure that it's coming from FBS.
Fantastic, thank you, Jelena. So I think that's all the questions we had.
Just to add to that, but it's interesting, right? Because the bulk hydrogels were also made in the presence of that same media, right? So it still has the FBS in there, but the structure of the material is not allowing cells to spread and interact. So my point is that the morphology still matters, right? Regardless of the cell binding moieties.
That's exactly right. And I think you've shown it nicely in vivo as well, with your microstructure gels having a much better tissue filtration and the absence of the fibrotic layer on the outside as well, which was also really interesting. Another question there potentially as well, but probably in the interest of time, we'll go ahead as well. So again, thank you so much, Jelena, for your presentation today. It was absolutely great to have you here and show some of your amazing work. I can definitely tell you're, you know, really one of the emerging leaders in Australia and also globally in the field. And, you know, congratulations on all your achievements so far. It's absolutely great to have you here on board. So to all the attendees as well, make sure to register and sign up for future webinars as well. We will have 2 more this year as well, which we'll announce a little bit later in the year. Also make sure to follow Jelena on Twitter. She's very active on Twitter and often very entertaining as well. Make sure to follow her. And of course, us on LinkedIn as well for any future events that we might schedule very, very soon. And lastly, before we wrap up the seminar today, we'll be also sending out a follow-up email to all the attendees, which will give you another opportunity to ask any questions, should you have any that you weren't able to ask us now. So I think with that, unless I forgot anything important, I would just like to say thank you again, Jelena. Thank you for everyone who attended today, and we'll make this presentation also available online on YouTube very shortly as well. And yeah, with this, I would like to wish everyone a nice day and evening, and hopefully see you soon in the next webinar. Thank you, everyone.
Thank you very much. Thanks for having me.
Thank you.